Good morning, students. Today we will have our 10th session, and we will be starting the knowledge and understanding and the skills regarding mycology, or the science dealing with the study of fungal cells and the different types of fungi. So, the title of our session today is Introduction to Mycology, and mycology is the study of fungi بالعربي الفطريات. fungi طبعا it inhibit almost all types of environment, and they prefer the environment that is moist, that contains humidity or water. So this is the best environment that suits the growth of fungi. That's why they are found in warm and moist environments. Uh, Fungi, they are found or exist in two fundamental forms, either as unicellular, that means one cell, and this we call it yeast, or multicellular in form of filaments, and this we call them hyphal fungi. So these are the most important two forms of fungi in the form of their morphology. Either is it a single cell, unicellular yeast, or multicellular, and this we call them in the form of filaments, and we call them hyphal fungi. There could be a combination between these two forms, and that's why for classification, to ease the classification, these are further classified according to their shape and the number of cells into molds, and these are usually hyphal fungi, yeast cells, and these are usually single celled, yeast like, and this is usually they represent or they replicate the morphology of yeast, that there could be some differences, or dimorphic fungi, that they exist in the form of unicellular, but again they can be filamentous at one stage of their life cycle. We will speak about different types, and we'll have different types or different species for these different morphological differences. When we come to speak about the medical importance of fungi for human as pathogens. So first, we are going to start to talk about the main components, or the main structure of a fungal cell. So fungal cells are eukaryotic. That is means different from bacteria, but they represent the animal and the plant cell. So they are all eukaryotic cells. But of course, there are differences between the fungal cell and the animal and plant cells. The common components of fungal cell, regardless if it is hyphal or yeast, they have common components that is found in all types of fungal cells. First of all, they have cell wall. So they have cell wall. This is, of course, a main difference between a fungal cell and an animal cell. Animal cell, it's an eukaryotic, but it doesn't have a cell wall. So here, the fungal cells, they represent the plant cells in having a cell wall. So the cell wall here, it differs in its chemical components. It consists of chitins. Chitin, it's a polysaccharide that forms like microfibril units, that fibers, small fibers that is connected together and form like a mesh or a unit that help in increasing the rigidity and the resistance of the cell wall. So chitin is the main component of the cell wall, beside other uh, polysaccharides, which is beta glucan, which is a polymer of glucose, and mannan, which is a polymer of mannose. It is also a saccharide. So all these contribute to the rigidity of the cell wall, because the cell wall, it gives the shape of the fungal cell, and at the same time it has the function of preventing osmotic lysis. When we say osmotic lysis, because fungi, as we mentioned in the introduction, they prefer the medium or the environment where there is water and humidity. So definitely there will be an osmotic pressure difference between the cell and the environment where the fungi lives. So this prevent the osmotic lysis, that the water can enter from the environment into the into the fungal cell, and prevent its lysis. So this is one of the important function of a cell wall. The other component is a plasma membrane, and this is also called lamosome or plasmolemma. It encloses the inner components of the cell, which is the protoplasm. When we say protoplasm, that means the protoplasm, that means the cytoplasm of the cell. So it functions as a membrane, and it's differentially permeable. That means it allows certain components, chemical components, to enter from the environment into the bacteria, while it prevents other. So that's why it is differentially permeable. That is means it prevents and it allows other chemicals to enter into the cell, fungal cell, according to the need of the cell. So the membrane consists of proteins and lipids, as is found in the plasma membrane of other eukaryotic cells in plants and in plants and animals. But there is a major difference between the different types of eukaryotic cells. In the fungi, the lipid is different. The lipid component or the chemical component is different from that found in the plant and the animal. In the fungi, the lipid is called ergosterol. So ergosterol is a type of lipid that is found in the cell or plasma membrane. While in the animals, it is found, the lipid that is found is cholesterol. And in plants, it's phytosterols. So the type of lipid found in the plasma membrane differs between the three types of eukaryotic cells. So in the fungi is ergosterol, and this is the main component of lipids found in the plasma membrane. Since it is an eukaryotic cell, that means the nucleus is very obvious and it is well developed within the fungal cell. So it has its membrane, it has a nuclear membrane, and it appears under the microscope as a granular mass. That means it has granules within the nucleus. And this definitely goes back to the component of the nucleus, which contains the chromatin and the nucleic acids. That's why it gives its granular mass or granular shape under the microscope. So, but the most important thing is that the nuclear membrane that confines the nucleus within the fungal cell It has pores. It has pores. So these pores allows or facilitate the nucleoplasm exchange. That means the exchange of material or organic material, like what happens in the process of uh, uh, transcription and translation of the genetic material. There is an exchange between the nucleus and the cytoplasm. So this pores allows the exchange of materials between the nucleus content and the cytoplasm of the cell. The nucleus itself, it has a dark, dense central area, and this is called the nucleolus. And the nucleolus is known that it contains it contains very high components of RNA, which is a ribonucleic acid. So it is part of the nucleus, but it appears under the microscope as dense, dark area within the nucleus. The fourth component is the mitochondria. So the mitochondria, it is found in the fungal cell, and it's usually found in the subapical region. If the cell it is a bit elongated, so it is found in the apex part of the cell. So if it is circular, it depends, of course, on the shape of the fungal cell. It's found in a certain area, and usually it is mentioned that this area it is where the budding or the reproduction of the yeast cell starts to divide, because we are going to speak later about the reproduction of the fungal cells. There are different modalities, but one of them is budding and our bur'um. So this budding process, it needs very high energy, and usually this energy is provided by the mitochondria. So where the budding starts, usually the mitochondria is found where this area it starts to bud. So they are different in shape and size, and present, as I mentioned, in usually in the subapical region. They have double membrane, as you can see here. This is the mitochondria. It has a double membrane, and at the same time, the inner membrane invigilates or invigs inside the center of the mitochondria. So, or it folds to the center of the mitochondria. So these are called, these folds, each fold is called cristae. Cristae is this folding, and usually they are flat in shape. We don't want to go into details about the inner structure, but you need to know that this is consists of two membranes, and the inner membrane folds to the inside, uh, forming what we call cristae. So these folds, they are usually irregular and flat in shape. The other component that is found in the fungal cell is the endoplasmic reticulum. So the endoplasmic reticulum is a highly vascular endoplasmic organelle found within the fungal cell, and it usually originates from the nuclear membrane. That means it is usually found very close to the nuclear membrane, because its origin starts from the nuclear membrane, and it's usually found in hyphal or filamentous fungi that has hyphae, like molds. So this endoplasmic reticulum is found in this type of hyphal molds. The other organelle is the or component of the fungal cell is the vacuoles. So vacuoles, these are spaces found usually filled with fluids, or filled with certain chemical um, extraction, like extraction, it's form of extraction process from the cell. So these vacuoles are usually found in old fungal cells, but they lack the young fungal cells. They lack these vacuoles. So and usually they give the as the cell gets old, these vacuoles increase in number, and they give the strength to the fungal cell as it gets old. So to keep its cell growth and its shape. Um, the other type of components is the inclusion organelles or granules, as we usually call them in the bacteria, granules. So these granules are found in the fungal cells, and they are glycogen granules, granules, and they could be also granules or vacuoles in the form of vacuoles that contain protein materials like pigments, like enzymes, like amino acids. But the most important thing is that the fungal cells they lack starch grains or granules, unlike the bacterial cell. And the other thing, they lack the pigment chlorophyll. So, unlike the algae or other type of cyanobacteria. So, unlike, of course, the plant, which contains plant cell, it's a karyotic cell and contains chlorophyll. So, so the inclusions are mainly glycogen, lipid, and protein materials, and this protein usually enzymes, pigments, other type of pigments like carotenoids, carbohydrates, volutin, granules. These are all present within the fungal cell. Volutin granules are usually found in yeast, and molds, they have more of the glycogen granules or vacuoles. So we move on to the structure that is specific to the yeast and the molds, because we already mentioned that the fungi, they exist in two fundamental morphological forms: yeast, single cell, or molds, hyphal or multicellular um, type of morphology. So for the yeast, structure of the yeast, and as we mentioned, this is single cell fungi. They lack hyphae, but they reproduce by budding. If this is a parent cell, it starts to form like a protrusion or um, protrusion to the outside, and this is the start of the budding process. And when this bud elongates and starts to increase in size, of course, this all the material that constitutes the bud is from the original or the parent cell. And when it starts to bud, the nuclear material of the parent cell replicates or duplicates, and the duplicate part or the replica of the nuclear content goes to the bud cell, where it separates and becomes a single cell. So this is how the single cell yeast they replicate. Of course, the yeast, it has all the common components we already mentioned, and but it shows how this they replicate by budding. They contain Golgi apparatus, they contain mitochondria, of course, the cell wall, vacuoles, and so on. So the vacuoles here, this we mean the storage vacuoles, the granules inclusion vacuoles. So you need to differentiate between the vacuoles that is found within the old cells, and these are usually excretory. That means the cell use these vacuoles to excrete what is not needed within the cell, but at the same time, they keep the structure and the strength of the old cell. But when we say that inclusion, that means inclusion vacuoles that or granules. We mean these are granules or vacuoles that use for storage of nutrients within the fungal cell. So the second structure is the structure of molds, and as we mentioned, molds are multicellular and filamentous in shape because they have hyphae, and they have all the components we already mentioned, the common components as mitochondria, cell wall, and so on. But the molds, because they have hyphae and filaments. There are two types of hyphae. We call them, one of them is vegetative and the other is reproductive hyphae. As we can see here in the diagram, that this is a normal hyphae. As you can see, it has a wall. It has a plasma membrane that invaginates into the inside, endoplasmic reticulum. It has vacuoles, but as you can see, the nucleus, because this is this hyphae could have separate walls separating cells, or they could be all basing or found in a single cytoplasm. So you can see, if you look into the hyphae under the microscope, you might see multiple nuclei within a single hyphae. So this is a mitochondria, a number of mitochondria, but certain hyphae, they have septates, or they are divided into single cells attached together, like here. So we have two type of hyphae, either with septates that separate the cells, or without septum, or cell wall dividing the cells, and they all the components, they pass, or they are found in single cytoplasm. So regardless of it is septated or unseptated, the hyphae are two types. We have the vegetative hyphae, and this is found usually in the medium where they live. It is found within the medium where they live, and this hyphae, they have like um, um, uh, uh, what we call a network. They have like small absorptive functions, and these, they help in the absorption of the food for the fungus. So this type of hyphae, we call it vegetative hyphae, because they help in the in absorption of food from the environment or the material where they live. The network of this hyphae, we call it mycelium. So they divide, and they give like branches, and these branches, they constitute what we call them a mycelium. And the other type of hyphae is the one that is protruding out of the environment, and this one we call it reproductive, because this is what we call a fruiting body. It contains spores, and once the spores, they disperse, and we are going to see this later when we speak about the different ways of dispersion of fungi and reproduction. So they contain spores. Once these spores, they move to other area, then they start grow and give the same shape as the original hyphae. So we have mycelium and the reproductive hyphae. This is the vegetative hyphae, and this is the reproductive hyphae. So usually this type of molds, they replicate, or they reproduce by dispersion of the spores, and then the spores, they can reproduce sexually or asexually. But we are going to speak about this more when we come to speak about the reproduction of fungal cells. So we move on to the classification of fungi, and the classification of fungi, as we mentioned before, they are representing plants. That's why in the early science of mycology, they were grouped under the plant kingdom, because they are eukaryotic, and they have cell wall, and that's why this were the main characteristics why they were grouped under plant. And that's why early in the science of mycology, they were of importance and of attention to the botanists, those who are dealing, scientists who are dealing with the science of plant origin. But as the knowledge and the technology and the identification processes and the development of microscopes further developed in the science of microbiology, in 1969, then Whitaker, as we mentioned before in the bacteriology, classified all living organisms into five kingdoms, namely Monera, Protista. Fungi was a separate kingdom, Plantae, and Animalia. So, since that time, the kingdom of fungi consists of eukaryotic organisms that includes three types of fungi: yeast, molds, and mushrooms. We are going to have examples for this, but let us first go into further classify modes of classification of this fungi. So the kingdom fungi are further classified based on different types or different modalities. The main classification was based on two moods: mood of nutrition, and the other type is the spore formation. So let us first talk about the based on the mood of nutrition. So if we speak about the mood of nutrition, then we have three types or groups of fungi. The first one is the saprophytic, and when we say saprophytic, these are the fungi that feed or live on dead organic substances. That's why you can find them in dead on dead bodies, or you can even grow them in laboratory media, because laboratory media is considered just uh, decomposed decomposed chemical compounds that is ready for the fungi to absorb. So these are mainly containing rhizopus, aspergillus, and penicillium fungi. The other type is the parasitic. Parasitic, that means tophaili, and these live on living, living, living organisms like plants and animals. And as you can see here in the photo, these are leaves of plants. There are two types, for example, of fungi, the Taphrenia and Puccinia. So they live on the leaves of plants, and they use the food found in these living leaves or trees for the benefit of the fungus. That's why we call them parasitic. Symbiotic, these are the mutual benefit between the living organism and the fungus. There are two more main types: lesions and mycorrhizae. The lesions, they have symbiotic association with algae, polyp, and usually they are benefiting each other. So here the algae and the fungi, they have mutually benefited. The fungi, they provide the shelter for algae, while the algae synthesize carbohydrates for fungi. So the fungi make use of these carbohydrates and store it within their cell as inclusion granules. Mycorrhiza is the other type of fungi that is symbiotic with have symbiotic relationship with plants, and as you can see here in the photo, it's they are growing on the roots of plants, and this helps the plant to increase the surface area of the roots and help in the absorption of the nutrients or by the plants. But at the same time, the plant provide organic molecules like sugar to the fungus. So the fungus make use of the absorbed sugar, um, provided by the plant for the benefit of the fungus. So there is a mutual benefit. Here, the lesions and the algae, as you can see, the algae are usually the one that is green in color, and the lesions that is are found here in association with the algae. This is a bark of a tree. They are living symbiotically in the on the bark of the tree, and they are cooperating or mutually benefiting from the shelter of the fungi provided by the fungi lesion, and the algae they provide carbohydrates that is that is of course by photosynthesis by the algae and provided to the fungi. So we move on to the other classification, which is based on the spore formation. As for the classification based on spore formation, so the kingdom fungi are classified based on the type of the spores produced by the different types of fungi. And they are classified into four groups. The first group is the zycomycetes, and these are found they are found saprophytically. They live saprophytically. That means on dead organic matter or decaying organic matter. And you need to know that usually fungi they have two reproductive processes, sexual and asexual. This is a normal thing, but the difference in the type of spores produced from the asexual process so that it can propagate sexually. So in zycomycetes, at this stage, you need to know that the zycomycetes, they produce sexual spores known as zygospores. As you can see here, it is produced by the fusion of two cells from different hyphae, and they give what we call zygospores. This is a sexual spore, zygospores. But the asexual spores are called sporangiospores. These are sporangiospores from the reproductive hyphae. We are going to speak, as I mentioned, when we come to the reproduction, we are going to speak more about the cycle of reproduction of the different types of these groups. So in the zycomycetes, what you need to know, sexual reproduction produces zygospores. In asexual, it produces sporangiospores. And this type of this group is the example for this group is a mucor. The other type of, uh, of, of a group of fungi according to the type of spores formation, what we call the ascomycetes. When we say ascus, that means it's the shape of sac or kiss, what we call sac. So these are also called sac fungi or asci, and these are parasitic or saprophytic. They can live both as parasitic or saprophytic. That means parasitic on living organisms, but they are living for their benefit. Saprophytic, that means they are living on dead organic matter or dead organisms. The sexual spores are called ascospores. That is means they are producing like ascospore, okay, from the sac, and asexual production is called conidiospores. So they are producing conidiospores. So the conidiospores here, as you can see, this is ascomycetes. They produce ascospores based on the sexual and conidiospores based on the asexual reproduction. At this level, as I mentioned, you just need to know the types of spores, not the life cycle or the reproductive cycle. We will speak about this more when we come to speak about the reproduction of fungi. Basidius mycetes, these are mushrooms and commonly called basidio, the spores produced from the sexual production are called basidiospores. The body that produces what we call this is the body that produces the spores. We call it basidiomycetes. That's why they are called the spores themselves, they carry the same name of the body that produces these spores, what we call basidiospores. In asexual, they reproduce by the spores called conidia. So the conidium is an asexual type of basidiomycetes, and as you can see, there is sharing. They are sharing the name of the asexual here in ascomycetes and the basidiomycetes, although there are different types of fungi. This type of fungi is called, an example of this is agaricus, and this is type of basidiomycetes. The fourth type is deuteromycetes, and they are this group, we call them imperfect fungi because they do not reproduce um, sexually. They just reproduce asexually by the formation of conidia or conidiospores. And this type is the trichoderma for this group of fungi. So as I mentioned, you just need to know for this step the type of spores, because this is the base of classification of the four types of fungi. Thank you, and I'll meet you on the coming session. Goodbye.